Our today's topic of our discussion is metabolism of bilirubin, or we can say jaundice. Jaundice is a, symptom. For understanding what is this bilirubin and the derivatives of the bilirubin, so first we need to understand what is cholesterol. So cholesterol is a molecule. It is a lipid soluble molecule. It is also sterol, and it is very important in the. It gives the fluid property of the cell membrane, and it also helps in the synthetic processes. Okay, so this is the uh, structure of the cholesterol. As you can see, these are the four rings, and on the carbon number two we have a side chain. Okay, and on the first carbon of the A chain we have hydroxyl group. Okay, so uh, so we will talk about the degradation of heme. So first of all, what happens that the hemoglobin which is present in the RBCs, it gets degraded by the enzyme called hemoxygenase, and hemoglobin it dissociates into the heme and the globin part. Now the globin part, it is basically a protein. Okay, so protein is basically, what is protein? Protein is basically a group of amino acids, right? So amino acids, they can be recycled for the production of hemoglobin. Okay, or basically the globin part of the hemoglobin. Okay. What do we have? Hemoglobin, heme and globin. So globin gets recycled and we have heme. Okay. So heme, okay. So after 120 days in circulation, the RBCs are taken up and degraded by the reticular endothelial system, and approximately 85% of heme destined for degradation comes from senescent RBCs. Senescent means old RBCs, which have become old. So the remainder is from degradation heme protein other than hemoglobin. Okay. So uh, now there is a system called as a reticular endothelial system, in which there are macrophages, uh, dendritic cells, and B cells, which are very important in our immune system. They are basically called as antigen presenting cells, and yeah. So this reticular endothelial system is formed by monocytes, like the macrophages. They are the derivatives of monocytes, and yeah, and they can phagocytose this heme portion of the hemoglobin. Yeah, and this rest are located mainly in the liver and spleen. Okay, liver and spleen, liver and spleen. Okay. So the degradation of heme is catalyzed by microsomal enzyme hemoxygenase, and the end products are biliverdin, which is a green pigment, carbon monoxide, and iron. Okay. So biliverdin, it's a green pigment. As you can see, biliverdin is a green pigment, and carbon monoxide, it is very toxic to our body, so it is again reduced to the carbon dioxide by the enzymes in the cytosol. And iron, now there is a very interesting theory about the iron, like if the body needs iron, then it is exported from the cells of enterocytes. Okay, and if it is not needed, then it is stored in the form of ferritin. Okay. See, that is what I have explained now. So free iron joins in intracellular level iron pool when there is a low demand for iron in the body. As well, iron is stored in ferritin. When iron demand is high, the absorbed ferrous iron is transported across the basolateral membrane into the blood. Now, formation of bilirubin. So, biliverdin is reduced, forming the red orange bilirubin, and its derivatives are collectively termed bile pigments. Okay, bile pigments. So if anyone asks us about what is bile pigment, so we can say that the bilirubin and its derivatives are called as bile pigments. Okay, and bilirubin in small quantity is in small quantity it can function as antioxidant. These are the colors of the degradation of cholesterol. After the cholesterol has been degraded, what do we get? Protoporphyrin of 9. What is this? This is hemin, biliverdin, and bilirubin. Okay. So the uptake of bilirubin by liver. So actually, this bilirubin is unconjugated form of bilirubin, which is lipid soluble. Lipid soluble. Okay. So this cannot be solubilized in the plasma, right? So we need a carrier protein called albumin. Albumin transports this bilirubin to the hepatocytes, and when it passes near the hepatocytes, it gets passage it gets absorbed into the hepatocyte via the process called as a facilitated diffusion. Okay. This means that constant movement of ion or a molecule from a high concentration to the low concentration via the carrier protein. Okay. This is called as facilitated diffusion. Okay. So, in this way, the bilirubin, that is unconjugated bilirubin, it enters the hepatocytes, and then it bind to the then it bind to intercellular protein called as ligandin. Okay, this is ligandin. Okay, okay. So, formation of bilirubin diglucuronide. Okay, so what have we learned till now? That the bilirubin enters the hepatocytes via the facilitated diffusion. Okay. Once the bilirubin is inside the hepatocytes, it gets conjugated via the two molecules of glucuronic acid. Okay. So this glucuronic acid, they are transferred to the bilirubin, and it forms a bilirubin diglucuronide. Okay. And this process is carried out with the help of enzyme called as a bilirubin UDP glucuronyl transferase. Okay. It's a very complex name. Actually, my friends, they pronounce it wrong. Huh. Okay, that's the very complicated bilirubin UDP glucuronyl transfer. Even I can't pronounce it right. Huh. Yeah. So this process is referred to as conjugation, and so bilirubin diglucuronide is called conjugated or direct bilirubin. Okay. So this unconjugated bilirubin, it gets converted into the conjugated form, and this conjugated form is water soluble, that is plasma soluble. Okay. Fine. So then secretion of bilirubin into the bile. Okay. So what have we learned? The bilirubin, it enters the hepatocyte and gets conjugated via the enzyme called bilirubin UDP glucuronyl transferase to the bilirubin diglucuronide. Okay. So this bilirubin diglucuronide, it enters the bile, um, gets secreted by the bile, and huh. So against concentration gradient means active transport. Active transport means with the help of ATP. Okay. So into the bile canaliculi and then into the bile. Okay. Unconjugated bilirubin is normally not secreted into the bile. Unconjugated means that only bilirubin. Okay. Unconjugated means only bilirubin, and conjugated means bilirubin plus the two molecules of glucuronic acid. Okay. So okay, this is not important. So formation of urobilins in the intestine. So um so this bilirubin diglucuronide is hydrolyzed and reduced by bacteria in the gut to yield urobilinogen, a colorless compound. Urobilinogen is important. A colorless compound. Okay. So most of the urobilinogen is oxidized by the intestinal bacteria to crystallocopilin, which gives species the characteristic brown color. Okay. However, some of the urobilinogen is reabsorbed from the gut and enters the portal blood. Okay, a portion of urobilinogen by enterohepatic circulation is taken up by the liver and then resecreted into the bile. Okay, and yeah. So what can we say about the urobilinogen? So urobilinogen, it's a colorless compound and it has three fates. Okay. First, it may get oxidized via the intestinal bacteria to the stercobilin, and this gives a feature of a brown color in the feces. And for second, it might get again into the liver via the enterohepatic circulation, and again be reused for the process of this formation of feces, like brown color of the feces, or it may like get reabsorbed into the blood, and in the kidney there is a bacteria C. Oh, sorry. So it's transported to kidney where it is converted to yellow C, yeah, yellow urobilin and excreted, giving urine its characteristic color. Once this is the diagram, you should you read this. It is very important. Okay, you please read it, and if you have a doubt, you can ask me in the comments. Okay. Now talking about jaundice. So jaundice is it is also icterus. It refers to yellow color of skin, nail beds, and sclera caused by deposition of bilirubin, secondary to increased bilirubin levels in the blood. It is called as a hyperbilirubinemia. Okay. Jaundice is usually a symptom of underlying disorder. So, there are three types of jaundice according to the type of bilirubin in the plasma. We have predominantly unconjugated bilirubin, hyperbilirubinemia, predominantly conjugated hyperbilirubinemia, and mixed conjugated. Okay, this is you can read this simple. We were talking about the types of jaundice according to the etiology. So we have three types of jaundice: hemolytic, hepatocellular, and obstructive. So in hemolytic, what happens that there is increased rate of RBC destruction. So increased hemoglobin breakdown to bilirubin in rest, that is reticular endothelial system cells, that is the macrophages, and this exceeds the capacity of conjugation in the liver. So if there is a no like there is no space for the conjugations, therefore we have unconjugated bilirubin in the plasma. Okay, and in the second type we have the hepatocellular jaundice. So what happens in this is that there is a, due to some pathology, there is a uh, destruction of the hepatocytes. Therefore there are not much cells in the liver to perform the conjugation, and therefore the unconjugated bilirubin is found in the plasma. Okay. And in the obstructive, what happens that all the processes like conjugation, then secretion of the bile, of the bilirubin is it is well marked, but the thing is that there is obstruction of the pathway of the bile from where it is excreted into the small intestine. It is obstructed. So therefore, the conjugated bilirubin, it cannot reach the this small intestine. So therefore, it again reabsorbs in the plasma, and therefore we get a high level of conjugated bilirubin in the obstructive. So in hemolytic and hepatocellular, we have unconjugated bilirubin in the plasma, and in the case of obstructive, we have conjugated bilirubin. Okay. And jaundice in the newborn, the majority of newborns, infants, 60% of full term and 80% of the preterm show a rise in UCB, that is unconjugated bilirubin in the first postnatal week. This condition is called transient physiologic jaundice. Causes low level of hepatic bilirubin, UGT. What do we know about UGT? It is called as a bilirubin, UDP, glucuronil transferase. Okay, and treatment blue fluorescent light phototherapy, which convert bilirubin to more polar and therefore water-soluble isomer. Okay, so this is what I had today for bilirubin, and thanks for watching. And if you like this video. Okay, and this is Abrar Pathan signing off. Bye.